Okay, let's get going on the CAR T cells. You've heard about these already from Dr. Jacobiak for multiple myeloma, and we are going to touch on myeloma as well. Thank you so much. So what are CAR T cells? And again, CAR T cells basically are based on the notion that our immune cells are very effective in treating cancer. Our immunity protects us against many pathogens, in particular viruses and bacteria. It also helps prevent cancers from developing and helps us in treating them. Most important in our cancer surveillance is our T cell lymphocyte population. These T cells are found all over our body, lymph nodes, spleen, liver, and the blood. And a deficiency of T cells is seen in many cancers. And there's a yin and a yang in the T cell population, and the suppressive T cells, are called regulatory T cells, are actually increased in many patients with cancers. So T cells in cancer are impotent in most cases. They fail to recognize and kill cancer cells. CAR T cells, on the other hand, are manufactured T cells that are engineered to wake up the immune system and help us kill cancer cells. So T cells do most of the work in preventing and eliminating a lot of diseases, not just blood disorders, but all sorts of cancers. And you've heard the commercials in which they talk about immune therapy for psoriasis or Crohn's disease, etc., and they always have a disclaimer that these therapies can cause lymphoma. Well, lymphoma develops because you're suppressing the immune system, which allows the abnormal clones that probably develop in all of us all the time to increase in our bodies and cause cancer. So there are actually two forms of engineered T cells that people are getting right now. The CAR cells, the chimeric antigen receptor cells, in which T cells are manufactured directly to attach to the surface of tumor cells, and the TCR cells, the T cell receptor cells, in which they attach to the tumor cells, but through their normal receptors, which have to do more with the insides of the cells. And because of that, we have to match these to your HLA system. And we heard about HLA for in the bone marrow transplant. Most of the TCR cells are HLA-A2 restricted, which covers about 25% of the population. So they're capable of recognizing things going on inside the cell, not just on the surface, which is important for certain cancers. We have TCR therapies now ongoing at our center, for example, for malignant melanoma and soon renal cell cancer with the NIH. So here shows the different strategies on the TCR cells. They're not a lot different. You basically attach the gene that you want to put into the T cells through a virus, either a lentivirus or a retrovirus. And again, what you put into the cell will determine whether it's a CAR cell or a TCR cells. So for the CAR cells, or chimeric antigen receptor cells, a virus carries a signal into a T cells that codes for a new antibody-derived binding to the cell surface that recognize normal antigens. Now, normally our T cells don't attach to tumor cells through antibody binding. Our B cells do, or antibodies do. Rituxan is an antibody that binds to the tumor cell and kills it. What this does is it attaches the T cell right to the tumor cell, and then the T cell can actually kill the tumor cell. So infuse these cells target molecules on the cell surface, which lead to their cell death. So here's the chimeric antigen receptor. We have a tumor cell here that has CD19 on the positive, and the CAR cell is manufactured to have an anti-CD19. So this anti-CD19 then attaches to the CD19 of the tumor cell and then is killed. How do we get the CD19 on the T cells, which are not normally there? We put the gene into the tumor cells through a virus. So here's a schematic of what happens. We collect normal T cells from the patient. We manufacture them, and this sometimes takes 3 to 4 weeks to do and quality control it, and then we infuse the CAR cells back to the patient. So they have been effective in acute lymphoid leukemia of the B cell type, non-Hodgkin's lymphoma of the B cell type, chronic lymphocytic leukemia, and you've heard already a little bit about multiple myeloma. The first effective therapies for CARs were in CD19 positive B cell acute lymphoid leukemia, mostly in the pediatric population. And as you can see here, first of all, they're not 100, 200, 300 patient trials. These are 20 to 30 patient trials. However, for patients who had refractory acute leukemia, you can see 90, 70, 83% of the patients went into complete remission. The toxicities we'll talk about in a moment, but again, very impressive results. So the first experiences were developed at the National Cancer Institute, and the results published in 2015, in which patients who got truly a complete remission, 80% of them remained in remission. And when you looked at all patients at risk, again, a small number of patients, granted, 20 patients, about half of them were alive at periods of 2 to 3 years. This drug, which is manufactured by Novartis now, is commercially available to treat children and young adults with B-cell ALL based on this trial that was published just a couple of months ago in the New England Journal. This drug has been available since August, in which 107 patients were screened, 92 were enrolled, and 75 were treated. This is the data, again, in these children showing approximately 50% of the patients alive and well at 15, 16 months. So the rate of relapse-free survival among patients with a response to therapy was 80%, and 60% at 12 months. Among the patients with a complete remission, 17 had a relapse before receiving additional anti-cancer therapy. So in many instances now, we do these treatments and then add on a bone marrow transplant or other maintenance therapy. Relapse occurred in 3 patients who proceeded to receive new cancer therapy for the emergence of minimal residual disease. However, again, based on this result, for this patient population that would normally have a survival of 20% or less just with a bone marrow transplant, now it's up to 50% early on, led the FDA to release this commercially for patients with B-cell lymphoblastic leukemia who are up to the age of 25. There are adult trials ongoing. We have one of them at our institution for patients with B-cell diseases. Results hopefully will be as good. We'll have to wait and see. The second area that is commercially available are the Kite Pharma, which is part of Gilead now, in using these cells, a different CAR, but in refractory B-cell lymphomas. And this also was developed at the National Cancer Institute. And I won't get into the details of how these are made, but again, here still the same specific antibody to CD19. So again, we collect the T cells, ship them to the manufacturing. They activate the T cells, transfect them with the virus that carries the CD19 into the cell. We expand the cells, cryopreserve it, and then ship the patient the product. They grow for 6 to 8 days, but in reality, it takes about 3 weeks to get these cells back. The first patients treated, about 30 patients again. Response rates, again, very high, 60 to 80 to 100%, especially for indolent lymphomas. Complete remission rates were a little bit less, in the range of 30%, and ongoing responses were seen in a small portion of patients. Now, any of you have ever had a PET scan? This is a radionuclide scan that determines disease activity, and all these black spots all over the body here represents disease. And after treatment, the PET scan in this particular patient with large cell lymphoma refractory who had failed an autologous transplant showed a complete disappearance of the disease. And here's a cut through the liver, and here's these other lymph nodes totally gone. And that can and does happen in these patients. So here's a U.S. multi-center trial that we participated in. Overall, in large cell lymphoma, 31% of the complete remissions. Transformed follicular lymphoma, 50%. So overall, 1/3 of the patients did well at a period of 6 to 12 months. And again, just showing here lymph nodes here. All this area here is lymphadenopathy, lymph node enlargement. It starts to shrink at 1 month. And here, 2 years later, it's totally gone. And some of these patients now are 2 and 3 years. This led again to the FDA approval. This was just published again in the New England Journal in December of this past year, and it showed overall about 1/3 of the patients are doing well. Now that means that 2/3 don't, and those are patients that don't have a response, and unfortunately the disease can go back very quickly. But those who have complete remissions actually do quite well. Now you can say, you know what, 30% is not that great, but indeed these are patients in whom the overall chances of survival would be in the 10% range, so a very poor risk patient population. And again, like we talked about for the AML, we're using this in heavily pretreated patients toward the end of their disease state. Using it a little bit earlier might indeed see an improvements in responses and better outcomes. The availability, several centers in Chicago are exploring these T cells for lymphoma and B-cell ALL in ongoing research trials. Our center has a trial for mantle cell lymphoma and adult ALL. The FDA, however, has licensed CAR T cells for commercial use in pediatric ALL, adult aggressive non-Hodgkin's lymphoma in November, and Novartis just got their approval a couple of weeks ago, which is the product for the pediatric ALL. These cannot be administered at community centers. They're currently only approved for by the companies for centers that have a FACT-approved bone marrow transplant centers. This has to do with the toxicity of these therapy. Before we talk about the toxicity, let's finish up for just a moment on the CAR T cells for multiple myeloma. We talked about this a little bit earlier. The BCMA is a B-cell maturation antigen, which is seen selectively restricted to plasma cells and mature B cells. It's not detectable on any normal tissues. It's nearly universally expressed on multiple myeloma cells and has validated efficacy in early trials. And you can see here the staining of BCMA on multiple myeloma cells showing that it does really hit these cells effectively. Now this is one of the strategies you heard about this morning, immunotherapy for multiple myeloma, again in which the CAR T cells attach to the BCMA. We talked about PD-1, we talked about daratumumab, and we talked about other vaccines already today. The first trial was done also again at the NCI, National Cancer Institute, all at low doses in a dose escalation. There were minor response. However, at the higher doses, more impressive responses were seen, including complete remissions with drug-refractory disease. Toxicities, which we'll talk about in a moment, are dose-related. So the higher doses, which we hope we didn't have to get to, are seen at the higher doses and not the lower doses. In some patients, there was also a prolonged suppression of blood counts because the drugs may affect blood count as well. And of course, like any new therapy, relapses do occur. We saw this already in which some of the patients did well long-term. My color here is more of an aqua color here with long-term remissions. Again, some patients end up with remissions and then relapse here, but as you got to higher and higher doses, the results got a little bit better. Bluebird's data, which we also heard about again, showing that at low dose, very few of the patients responded, but again at high doses, more of the patients responded. So here's kind of a summary of all the data that's out there so far. The NCI, the University of Pennsylvania, which works with Novartis, the Bluebird, and the LCAR studies. Again, as you can see here, the number of patients that have been treated, here's 12, 9, 28, and 40. So we certainly can't claim that we're curing cancer because we just haven't gotten enough data. This is data from literally the last 12 months. Overall response rates, 42, 33, 94, and then here's the Chinese data that sticks out as kind of a very high response rate, and that obviously needs to be verified. This is not subject to US oversight and criteria, whereas all these other ones are. So the downsides of CAR T cells have to do with the toxicity. When you give somebody a very potent immune cell and just throw that into the body, and there are lots of tumor cells that it starts to attack, you end up with side effects. And here's the pediatric trial showing the cytokine release syndrome occurring in 3/4 of the patients, almost 80%. Grade 4 toxicity in 25% of the patients. We'll outline what that is in a minute. 40% of the patients had a neurologic event. 43% had an infection. 35% had fever associated with neutropenia associated with the treatment that we give the patients. And about a third of the patients still had low blood counts even 28 days after therapy. And tumor lysis, the cells can sludge up the kidneys, was a very uncommon phenomenon. So the cytokine release syndrome is again something that's seen in 3/4 of the patients. When you infuse the cells start to kill. That releases chemicals in our bodies that cause fever, drop in blood pressure, and shortness of breath. Whenever we get a virus infection or whenever we get really toxic from infections, viruses, etc., we develop fevers and occasionally hives, shortness of breath, etc., rashes. And that's due to chemicals in our body that the virus induces, if you will, based on the infusion of, in this case, the T cells or the virus itself that causes our sore throats and pneumonias, etc. So it's due to reaction of the CAR T cells with the tumor cells. Again, 3/4 of the patients got it. Median onset was 3 days. That's why this therapy needs to be done in the hospital. And it lasted 8 days. What happens to these patients are that the blood pressure drops. 47% of the patients had to go to intensive care unit for a median of 7 days. 25% were treated with high-dose vasopressors, which means that blood pressure medicines to maintain your blood pressure, as you sometimes see in septic shock. 44% required oxygen supplementation. 13% of the patients ended up on a ventilator, and 9% underwent kidney dialysis for renal failure. 37%, however, received a drug that manages this a little bit better. So we're seeing that these were very early on toxicities, and we're actually seeing reductions, but still patients are at risk of developing these. Neurotoxicity is the other major complication. We don't totally know what causes this, but it can consist of confusion, seizures, and altered mental status and coma. The most common we're seeing in 11%, that's encephalopathy, confusion. Some of the patients stopped talking for a bit of time. Confusion, delirium, tremor, somnolence, and one patient had a seizure. Among the grade 3 or moderately severe toxicities, 50% resolved within 10 days and 75% resolved in 18 days. So again, it gets down to a question: if you have a patient, especially a pediatric patient, who has a benefit of a therapy in the range of 75 to 80%, do you not give it because of toxicities? No, you do give it. You watch the patient very carefully in a very structured environment. You work with the toxicities. You try to ameliorate them. And as we develop new therapies with the CAR T cells, modifying the CARs, giving them a little bit different strategies, we hope that over time we're going to reduce these toxicities. So this is really basically groundbreaking, very, very, very early work in which we're seeing toxicities, but also a very gratifying response. Now, some of you may know that when these first came out, the biggest thing that was in the media, besides the fact that they were effective, was the cost of these. The Novartis drug cost $475,000 for these cells. But when you think about this therapy for the pediatric age population or young adults, we do have what we call for therapies quality adjusted life years that we use for things like kidney dialysis and blood transfusions and surgeries, etc. And we feel that if there is a threshold between $50,000 and $150,000 for QALY, that it's useful therapy, and even at $475,000, considering the number of lives saved and potentially the value, it's only about $46,000. So it is a cost-effective therapy for this patient population. The adult patients who are getting the Kite product is a little bit less. It's about 1/3 less cost. And recognizing the less effective therapy and no long-term outcome, the cost-effectiveness ratio is 136, not 46. So it's coming up to the upper limits of quality adjusted life years. So we need to watch that over time and make sure that we're going to be able to reduce the toxicities, reduce the other costs, and potentially as new companies get into the market, we'll end up with less expensive therapies. So in conclusion, CAR T cells can, in approximately 50% of pediatric ALL, who are refractory to ALL be curative? And that's kind of a out there statement, but yes, it can be curative. We have some of these kids out 3 and 5 years now who otherwise, if they had just one cell, would have had a relapse long ago. CAR T cells in approximately a third of adult large cell lymphomas can potentially be curative. We don't have as long data, but it looks very promising. 100% of the patients will get the toxicities as well as prolonged bone marrow suppression, and we need to make sure these patients are very carefully monitored and observed. Efforts are ongoing to make these cells less toxic and more effective, and I think that that's certainly going to be possible over the next several years. So I'll stop there and answer any questions you might have, and then after our last talk, we'll have all the docs come up and we can each give our thoughts and comments on this novel therapy. Yes, sir, in the back. Well, transplants are a lot less expensive, and I think, again, many of the patients that have been treated so far on these trials are either not transplant eligible because they're not responding to therapy or they have failed a transplant. So this currently is being tested and being efficacious after transplant failure. So you can think of it, if it works when the transplant fails, could it be used instead of transplant? And actually there's a study that's just starting in which patients will be randomized between CAR Ts and transplant to see what is the role of each therapy. Know also that we're making advances in the management of patients with transplant, in which we're giving patients now, as you heard, maintenance therapy after transplant, which does, at least in Hodgkin's disease and multiple myeloma, improves our outcome. So transplants, the chances of significant toxicity that I've described are a lot less. It's rare for a patient to have the toxicities that you would see with cytokine release syndrome or neurotoxicity at all. The major side effects of transplant are nausea, vomiting, hair loss, and loss of appetite as being the major toxicities, and infection and bleeding are very, very uncommon. So none of those really severe toxicities. So if we ultimately see that they're equivalent, I think transplant will still win. If there's a 20% or higher improvement in an outcome for those earlier patients, then I think we might shift away from transplant and onto CAR Ts. So we don't have the data. So it was tested in pediatric population. Now we're testing it in adults. The preliminary data does show that there's some benefit. Whether it's going to be as good as kids remains to be determined. Yes, one more. More or less. An independent organization took the data, and they are an organization that does this type of analysis, and they do it for the federal government as well as the insurance companies. So you could be cynical and say, okay, how much can I charge for this based on quality of adjusted life years, and say, I'm going to come up to the edge of that and make sure that I'm within that, and figure out then retrospectively how much I can charge. I don't know if they did that or not, but this is an independent organization that actually does that. It's not the drug companies. It's very uncommon. Sometimes it takes a long time. It may take a month or so, and it would not be rare that a patient would recover and have to go to rehab. But again, if you have a patient with refractory lymphoma and the disease is gone, going to rehab for a few weeks and then living 5, 10, 15, 20 years is worth it. but that's only a third of the patients. So worst case scenario is you develop the toxicity and you don't go into remission. And for those patients, it is definitely a struggle to survive. And indeed, some of those patients may have had other options that are less toxic. And they may actually have done well for a longer period of time with some of those less invasive therapies. I think all of us think that this therapy, using it at the time when patients are truly toward the end of their disease, is not the right way to go. But when you test things like this, you do take those patients because they have, while they have a lot to lose, just like all of us do, they have less to lose than somebody who could be cured with just 6 months of outpatient chemotherapy. One last question, and then we need to move on to the last talk. Sorry. No. That's a completely different therapy. That works inside the cells. It's like the genetic therapy in my AML talk. It's like genetic therapy for lymphoma, and that's an injectable drug that's given outpatient. It's not cells. It's not your cells being given back to you. It can potentially accomplish the same thing. It's approved for follicular lymphoma. It's not approved for large cell lymphoma, which the CAR T cells are. Okay, let's move on. And again, there'll be plenty of time for additional questions. I want to bring in Patrick Hagan, who just got in after doing rounds. So he's doing double duty today, seeing patients in the hospital, and then while we've all been having a good time here, and now we'll be coming and talking to you about AML therapies, transplant versus no transplant. Patrick?